Hello, kiddies. My name is Professor Van Hornbottom. My friends call me Professor Van Hornbottom. But you may call me Professor Van Hornbottom. My assistant Arnold. Arnold. Yeah, yo, Professor. Get down. Get to the chopper. Arnold, you get out. You're messing up the show. I'll be back. We're here today to talk about the scientific method. I am a guest speaker for your teacher, Mr. Miller. What a guy, isn't he? Isn't he terrific? The scientific method is used by scientists to support or disprove a hypothesis. How do you know if the hypothesis is a good one or not? You use the scientific method. It's the only accepted way to do this. All scientists do this. They follow the steps, but they are not always followed in the same order, you see. The scientific method has 7 steps, although some might say there's less, some might say a few more, and they can be done out of order again. But first, usually you do observation. You do observation, and then you come up with a question. Well, why is it like this? What can we do? What kind of problem do we have? Then usually you do more observation, actually. And then you come up with your hypothesis. This is the possible answer. It's an educated guess. And remember, educated means that there is some sort of evidence, some sort of scientific fact behind it. Then you come up with your experiment or some sort of research or make more observations. This is where you do the testing. This is the testing of the hypothesis. Then you analyze the results, and then you come to your conclusion. And then you write up that conclusion and you communicate the results with others. They must know. They must know how smart you are. They must know, and you must share. That is all what science is about. Which steps of the scientific method are missed here? She misses a couple of steps. See if you can tell which ones and write them down here at the bottom.
The scientific method is organized common sense. Most people will tell you it has 5 major steps: observations, hypothesis, tests, results for those tests, and conclusions.
Biologists like these use the scientific method to systematically increase our knowledge of the world.
That was a good movie, no? One of my favorites. Should win an Academy Award. Oops. Now we go on to the first part of the scientific method. You make the observation. You observe something in the world around you using your senses. I wonder why. It usually leads to the next part, which is a question. See the little fellow here? He's asking a question to himself. Why is it like this? You ask a question about what you observe. And you state this problem or question. Here's the question this little fellow is asking. Which color of light do plants prefer? Then you would usually do more observations, but we are not putting that slide up again. You would make more observations and come up with your hypothesis. You predict what you think the answer to your question might be. You must research it first. That's the research or the observations we are talking about. This fellow has come up with a possible answer. Plants prefer the color green because they are green themselves. You should write this down. Never put I think or I believe, even though right here it says you think. You never write this. You never write that in your hypothesis. You state it as a fact. Scientists state facts. This is the way it is. It will be supported or not supported. Next, you would test your hypothesis. You could do an experiment. You design a way to test whether your hypothesis is correct. Or you do some research. Sometimes testing a hypothesis requires more observations or research. The test, it must be something that you can measure. You must be able to get some data from it, some numbers. Down here it says, must be measurable, have numbers. You make sure that you record or write down. You must write it down in a data table or a chart somewhere. Next, you must analyze the results. The best way to do this is to record the data and then make tables or charts or graphs so that you can look at it and make sense of it. You go, why is it like this? Then you come up with your conclusion. After completing it, you should analyze and look at everything, and then you write up your summary of the experiment. What happened? Was your hypothesis supported or not? You never say my hypothesis is right or my hypothesis is correct. You say it is supported. Underline or highlight that. Supported or not. Never right, wrong, or I screwed up, or I blew it. Nothing like that. Why did this happen? You should always in the conclusion write, why did you get these results? What did you learn from the experiment? What would you do next time? Most important is that you think deeply. You're always thinking about this. Spend time. Your conclusion should be long. Lots of information here. Look at this fellow. I don't know why he's scared of the explosion. In science, we always have explosion. I explode my lab every day. The scientific method. Notice how it always says the scientific method up here. We are talking only about the scientific method. Communicate the results. Share your results with the world. This allows others to learn from your work. Science is about sharing and helping other people. They can learn from your work. They can test it themselves and see, is it good? Are you right? Does it support the hypothesis or not? Share what you know. That is how you win the Nobel Prize. Like I have so many. I'm wearing them all around my neck right now. Oh, they're lovely. Okay, here's the next movie. I'm going to go ahead and start. It's not working. Arnold! Yeah! What? What? The movie's not working. Get the movie started. All right, Professor. Oh.
Mosquitoes, carriers of life-threatening disease, began to increase despite pesticide spraying. The biologists began their work.
First, they use a process of selecting a problem that they want to work on. That has to be something that is practical, they have the background, and then they begin to work on that problem. They ask a question.
Why were the mosquitoes increasing, asked the scientists, when pesticides should decrease their population? Their guess? The mosquitoes were becoming resistant, immune to the poisonous spraying. A guess based on some knowledge is called a hypothesis.
You make this hypothesis, which is an educated guess. Not true, it's an educated guess. But the game you play is this. I assume that the hypothesis is true. If the hypothesis is true, these deductions follow automatically. Then you test those deductions by observation and experiment.
The real key to using the scientific method is to come up with testable deductions from the hypotheses. A really good scientist, a great scientist, uses very insightful guesses for their hypotheses, but also they're exceedingly clever with the way they test those hypotheses.
The scientists devised a way to test resistance to a pesticide. They discovered a much higher concentration of an enzyme in the mosquitoes that were surviving. This natural enzyme in their bodies made them into super mosquitoes, resistant to chemical spraying, while others were susceptible. Now the scientists asked, did the pesticide actually cause resistance to develop?
You'd have to come up with a way of testing the original population before the pesticides at your control, and then you compare it with a much later population. And if there's a difference between the two groups, then that difference is because of the one variable that was there, the pesticide.
Scientists use control groups to ensure that they test only one variable at a time. For example, they tested mosquitoes from an untouched area and found that the insect's enzyme level was normal. In areas that remained pesticide-free, resistance did not develop. In areas treated with the pesticide, the mosquito population changed. It's important that scientists confirm their results.
Verifiable, repeatable information is the basis of scientific statements. If I say something about whatever aspect of nature that I happen to be studying, other people with the same training, with the same equipment, the same ability should be able to repeat those observations and come to the same conclusion.
Scientists showed that the mosquitoes had indeed developed a resistance to the pesticide, so spraying with that particular chemical was useless. The hypothesis was proven to be correct through verifiable data, the last step of scientific method. But just because a hypothesis is correct does not mean that science stops there.
In the case of facts in science, we do not regard those as ultimate fundamental truths. They may behave as though they are.
The problem is that as we get better tools and better ways of observing, we actually see things a little bit differently. So facts change. But that's actually considered the progressive nature of science, the fact that it doesn't stay the same, but things change.
So what we try to get are the best explanations that we can get for any phenomenon, and reach the point where this is true beyond all reasonable doubt. And we can work on that.
The fact is not important in and of itself, but as the fact points toward bigger ideas and bigger concepts, they help us make sense of our world, and that's where the theory comes in.
The way I like...
Well, that was quite a movie, huh? Lots of information. Super mosquitoes, very scary. Now that was a lot of information. Were you taking notes? You should take notes on all movies. Do not forget. Take the notes. Notice that the woman, she made one mistake. She talked about the hypothesis being proven correct. This is not the way you would say it. You would say the hypothesis is supported. The hypothesis was supported by the data. Now you must answer this question. You can go back and rewind. This is one of the nice things about the movie. You can sit here and watch it again and again, and you can go back and go, oh yes, this is what I missed. I must write this down. I can write more notes. I can answer this question. Why is it important to have a control group? It has something to do with being able to compare two groups together. You can be able to compare the two groups and make sure that the one thing that you change that's what's different. Go back and listen to what they say, what the girl says about this. Okay? We must go on. I hope. Here we go, the lab report. This is how you write up the lab report. You've already done one. Mr. Miller says you studied sewer maggots. Very interesting. I have heard of this creature before. The title, you would always do a title, centered and underlined. You are going to be doing this with your helicopter project, I understand? Then you must have your question. What color, here's a question, an example. This is just an example. What color of light do plants prefer? What is going to make them grow is what the question is. What's going to make them grow best? You must then come up with your hypothesis. You would do some observations, maybe some research. Maybe you have knowledge of plants as being a young kiddo. You might know plants prefer green light, you might say, because they themselves are green. That is a logical hypothesis. Plants are green, so maybe they like the green light. I know that light, you can have different colors of light. In fact, white light is actually made up of all different colors of light. So maybe they'd use the green light of the white light. Then you come up with your prediction for your experiment. Here is your if-then statement. If I do this in the experiment, then this will happen. If I put the plants under the different colored lights, then the plant under the green light will grow the most. Notice that it matches my hypothesis. The green light is going to help the most. So I would say, then the plant under the green light grows the most. I wouldn't say the plant under the blue light because that was not my hypothesis. Remember? My hypothesis. Plants prefer green light. And I always have a because. Don't forget about the because. You should underline that part. And don't forget about the if-then. You should circle or underline if-then. This one describes your experiment. Notice this is what I'm going to do. I'm going to put plants under different colored lights. Then this will happen. So now I know what I'm going to do in an experiment. You must figure out your experiment before you do a prediction. Now, I must figure out variables. If you remember correctly, Mr. Miller, that smart guy, Mr. Miller, he talked about having that variables are different things in an experiment that can change. These are the things that can change. You can circle that, right? Things that can change right here, right there. There's the independent variable. This is the variable that you decide upon. I decide it. You are trying to see if it changes or affects the results. In this case, the color of light. That is the thing we will be changing. We will be using different colored lights. Oops, I made a snake. I hit too early. So we have our different color of lights. The independent variable, the thing we are testing. Then we have the dependent variable. It depends on this variable, the independent one. It is what should change because of the independent variable. It depends on the independent variable. In this experiment, it is the height of the plant. Notice it is the thing we can measure. Underline, we measure it. This time we are measuring the height to see. A good healthy plant will grow tall, no? Whoa, hold on. What is this? Hold on, whoa. What if the plants aren't all the same kind? What if I forgot to water one of them for a couple of days? That would not be good. What if I have one next to a window and the others are all away from the window? These are all examples of variables that need to be controlled or to stay the same. Controlled variables, the book calls them constants, that book that you have. They are things that can change in an experiment but wouldn't be fair. If it was different for the different groups, it must stay the same. Controlled variables, that's what we were just talking about, controlled variables. These are things that must stay the same, or you'll never know if they were the reason for the results. You want only one variable that we are testing, right? The independent variable. No others. You can only have one independent variable, one thing changing. Maybe the plants grew taller because it got more water, not because of the color of light. Not controlling them can really mess up that experiment. I have done this a few times. Before I won my Nobel Prize, of course. The amount of water, kind of plant would make a difference, right? You couldn't have a cactus here and then another type of plant there. A dark room are some of your controlled variables. You have dark or light room. Don't forget, we must have the control group. It is different from controlled variables. This group, the woman talked about it in the videotape, this group would have white light. It is the group where you would not control the color. So you can compare your experimental group to it. It is the thing that we do not change. I believe in the sewer maggots one, your control group was just the plain water would be the control group. You need to have one control group of at least 2 plants. The more the better. We'll talk about why. Then you have your experimental group, or groups. There may be more than one experimental group. In this case, there's many. This is the group that you apply your independent variable to. Each subgroup, this is the groups that I was talking about, of at least 2 plants. More is better. Can you guess why? Why don't you write why? Why is it better to have more? Write that up here. Mr. Miller talked about that in some classes. Why do you think it's better to have more plants? Example, one group of plants for red light, one for blue light, and one for green light. Now we could do more lights, different colors, right? There's yellow light and purple or violet light, but we are just doing these three: red, blue, and green. And then the white light, which does not have any changes. That is our control group, yes? Then we have your materials. You should always list your materials. Just like you list the variables, you would list the materials. We have 4 groups, at least 2 plants, colored lights, white light, which is not really colored light, red light, green light, and blue. You would have a ruler. What is a ruler for? Oh, for measuring the plants. Make sure you plan ahead of time. 16 of the same size pots. 2 bags of the same soil type. Notice I have 16. I'm exact, very precise. Bags of soil, mixed are the same. Water. And don't forget your data table. You must always record. Record your data. Then you have procedures. You must follow the steps. What are the steps of your experiment? You will always write this down in your lab. If it's not done for you, you must write them down. Number 1, I place 4 groups of plants in a dark room. Then I measure the height of each plant before beginning the experiment. I must put this in my data table. This is the starting point, right? I must see how much they change. The dependent variable, how much higher they get. Then I place a different colored bulb over each plant. There's the light, right? I don't put them in a lit room because I want to have it all discolored lights. Then I allow each plant to grow undisturbed. I don't do anything different to each one for 2 weeks, but I make sure I water all of them exactly the same. That is a controlled variable, remember? Then I measure the ending height. How tall did each plant grow? And I calculate how much. Then I put it down on my data table, then I graph it so I can easily see this one grew this much, this one grew that much. This is the comparing, the analyzing those results, right? Analyze the results. There they are. These are my results. I'm going to go ahead and analyze them right now. I'm analyzing. Analyze, 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 analyze. We have the light color. Oh, the white light did pretty good. Red light, not too bad. Green light, not good. Very short, did not grow tall. Notice I put over here, average height, very precise titles. Average height in plants in centimeters. I say that how much that is so that we know these are centimeters over here. Down below here, I have what my different groups were. The control, the white light, the red, the green, and the blue. Down here I have light color. What color is the light? Notice the blue grows really well, but the green not so good. Does this support or go against my hypothesis? That is what I must be thinking about. One thing missing here is a title overall. It should say, height of plants are under different colored lights. Some sort of title tells what it's about. I'm missing that, not good. But here, down here, you've got your, this down here is your independent, or your independent variable. Right, this is the thing that we tested. The different colors of lights going to be down here. This is the dependent variable, the thing that we measure goes on here. This is a bar graph. There are different types of graphs you can do. Bar graphs, very good for this one, very good. For your helicopter project, I would do bar graph. Do bar graph. Then we must come up with our conclusion, the results. In this case, the results did not support my hypothesis. Notice I don't say my hypothesis is right or wrong. The results did not support my hypothesis. The plants grew taller under blue light. They actually grew the least under the green light. One thing is missing from here. Can you guess what it is? It's data. We're supposed to have how much. Bad, I did not put that in there. We should have, under the green light, they only grew about 2 centimeters, a little bit more, right? A little more than 2 centimeters, 2.2 centimeters. Under the blue light, they grew 5.2 centimeters. That should be in our answer. Under the plants grew taller under the blue light, 5.2 centimeters. They actually grew the least under the green light, 2.2 centimeters. Light color, this is what I was also in my conclusion. I say, what happened? Light color does affect the plant growth. Look, blue light provides the most energy, which helps it grow. After doing more research, I discovered that plants appear green not because they use green light, but because they reflect the light. The light bounces off. Green light comes back to our eyes. If they reflect green light, they cannot use it for photosynthesis. You can do research too on your helicopter project and put that in your answer. Next, I would like to research the effect of light intensity, how much light there is on the plants. That is what I want to do next. You should talk about that. What would I do next in this experiment? What's the next step? Oh, that is the end. Thank you very much. I appreciate your listening. You seem like nice little kitties. I look at you out there. You're looking very focused. Do the same thing with Mr. Miller. What a great guy, I tell you. And don't forget, Arnold. What? I want to say goodbye to the kids. Arnold, get out of, get away. Bye, kids. I'll be back.